Hello everybody. So in this video we're going to start by talking about how mitosis is different in plant and animal cells. Then we're going to talk about how the cell cycle is regulated, and finally we're going to end with a brief discussion of cancer. So first, if we go ahead and take a look at these pictures on the slide here, these photos are showing what mitosis looks like in a plant cell. So you can see this looks very similar to an animal cell where we have our chromosomes condensing during prophase, starting to line up during prometaphase, actually lining up down the metaphase plate during metaphase, separating at the centromere in anaphase, and finally having two unique nuclei during telophase. However, because we're in a plant cell here, we can't do cytokinesis quite the same way as we could in an animal cell. So we can't pinch off the cell membrane to create two separate cells, but instead, in a plant cell, we form a new cell wall called the cell plate in between my two newly created nuclei, thus creating two new plant cells. So if we compare an animal cell division with a plant cell division, you can see after telophase, when animal cells undergo cytokinesis, a contractile ring basically pinches and gets shorter to allow the cell membrane of that large cell to become two smaller cells. However, because of the cell wall in plants, this pinching or this formation of two new cells can't happen the same way. So instead, during cytokinesis, this new cell wall forms, and that newly forming cell wall is called a cell plate. Basically what happens is vesicles will bring cell wall building materials to the center of the cell and basically assemble a new cell wall in between the two newly developing nuclei. So that's a little bit about how cytokinesis is different in plant and animal cells. So now we're going to talk a little bit about how the cell cycle is regulated. So let's talk about this experiment that we have in front of us right here. So in this experiment, basically what scientists did is they took two different cells that were in different parts of the cell cycle and basically combined those cells into one larger cell and looked to see what happened. So you can see when they took a cell that was in S phase and they took a cell that was in G phase and fused them together to make a larger cell, the cell immediately started S phase, even though one nuclei from one of the original cells had not finished going through G1 yet. Similarly, when they took a cell that was starting mitosis and took a cell that was in G1, when they fused these two cells together, both cells started mitosis, even though the cell on the right was not ready for mitosis yet. So if you look, the DNA in this particular cell hadn't replicated yet, so this cell was not ready for mitosis. However, it started mitosis anyway. So what this suggested was that there were signals inside the cell that were signaling the cell, telling it what part of the cell cycle it should be in. So when the two cells fused together, the intracellular signaling molecules inside one cell also signaled the second cell, telling it to start a different part of the cell cycle. So the interesting part of this experiment is that the takeaway was, hey, there's something inside the cell that's controlling when the different parts of the cell cycle occur. So if we take what we learned here and take what a lot of other things that scientists have learned throughout the years, what we know is that there are different checkpoints in the cell cycle that can be regulated. So if we look at the whole cell cycle here, there are certain parts where proteins will decide if the cell is ready to go on to the next part of the cell cycle. And there are three important checkpoints that we have to know about. There's one at the end of G1, there's one at the end of G2, and then there's one kind of in the midpoint of mitosis. So these are the three main checkpoints that we're going to be focusing on here today. So as I alluded to earlier, these checkpoints are controlled by different proteins. So one really important protein for controlling these checkpoints, it's called MFP, and MFP stands for maturation promoting factor, and this is a particular protein that's responsible for the checkpoint that's found at the end of G2. And what this protein does is that it starts the phosphorylation cascade, which means it's probably a kinase if it's going to be phosphorylating something, that will initiate the steps of mitosis. So if we look a little bit more carefully at MFP, what MFP is like I said, a kinase molecule. It's called a cyclin-dependent molecule. A cyclin-dependent kinase molecule. And this protein is coupled with another protein called a cyclin. So these two things together make up the MFP maturation promoting factor. So the way these cyclin-dependent kinases work is that inactive version of these kinases are always present in the cell. However, when they bind to these molecules called cyclins, they'll be activated and thus they'll be able to phosphorylate whatever their target molecule is and thus get a reaction in the cell. Now, cyclins are named cyclins because they only are present in the cell at certain parts of the cell cycle. So the one half of the this complex here is always present, this inactive kinase portion, but the other half, the cyclin portion, is only present during particular parts of the cell cycle. So let's talk a little bit more about these cyclins. So like I said, cyclins are protein molecules that are made at specific points in the cell cycle. When they're present, they'll bind to the inactive CDK and basically allow the CDK to be active and phosphorylate whatever target proteins it phosphorylates. Then once the cyclins are not needed anymore, they are actually broken down, so they degrade, and then when they're needed again, and the next time the cell cycle occurs, they'll be made again anew. So you can see here, so this is an example of a CDK that looks like it's active during G1. So you can see the CDK is inactive for most parts of the cell cycle, and then at some point the cyclin gene is transcribed into mRNA and then translated into the cyclin protein. That cyclin protein will associate with the kinase molecule here, making it active, allowing it to phosphorylate whatever it has to do to signal the end of G1, and then after it's used, it's broken down again and the cycle begins anew. So now if we look at MPF, which remember is the particular factor that promotes the signal at the end of G2, it's the factor that allows mitosis to happen, you can see that as this particular cyclin cycles and becomes more present in the cell and then broken down and then more present in the cell and broken down, you can see the activity of MPF is only high when this cyclin is present, because that's when the cyclin will bind to the kinase and allow this protein to phosphorylate whatever it's going to phosphorylate. And like you can see, this happens during the mitotic phase, during mitosis, allowing the steps of mitosis to occur. So again, looking at a similar picture to one we looked at earlier, you can see the inactive kinase molecule is always present, no matter what part of the cycle we're in. The cyclin is produced when it's needed, and then when the cyclin is at a certain concentration, it's able to bind to the kinase itself, making this MPF factor, allowing us to pass the G2 checkpoint and start mitosis. And then once mitosis has been initiated, the cyclin gets degraded and the cycle happens again. Now, in general, that gave you an example of how a cell receives the go-ahead to pass a checkpoint and move on to the next phase. However, if a cell does not receive the signal to go ahead, basically what will happen is the cell will enter a non-dividing state, which we call G0. It's important to note that G0 is not like some separate location in the cell. This just means that the cell is not undergoing cell division. It's not preparing for cell division or dividing actively, and just going about its normal functions without activating the dividing process. So if a cell does not receive that go-ahead 0, we say it's in G0, which is a non-dividing state. So now let's take a look at another checkpoint here. So we also have another checkpoint that's basically in the middle of mitosis. And what happens here, if the spindle fibers are fully and properly attached to the kinetochores on the centrosome of the chromosomes here, then proteins will be produced that will allow the cell to pass this checkpoint. However, if chromosomes are not correctly attached to the chromosomes here, then the cell will fail to pass this checkpoint and their progress will basically stop there. So let's do a summary slide now where we look at all three of the checkpoints. So if you kind of go along, so after G1 occurs, the first major checkpoint basically makes sure that G1 has happened appropriately. So to pass this checkpoint, cells have to be of adequate size, there have to be enough nutrients in the surrounding environment to support 2 new dividing cells, we need the appropriate growth factors present, and DNA needs to be undamaged. So if all of these things are true, particular proteins will be produced that will allow the cell to move into S phase. The next major checkpoint comes at the end of G2. This is the one that's controlled by MPF, the one we discussed earlier. And the main thing this checkpoint is looking for is to make sure that chromosomes have replicated correctly and DNA is undamaged. In other words, it's checking to make sure the S phase has happened properly. And if these two things are true, the cyclin that we discussed earlier will be produced and MPF will be active, allowing the cell to move into mitosis. And then finally, the last checkpoint we talked about happens basically in the middle of mitosis, and it checks to make sure the chromosomes are all attached properly to the spindle fibers. And it is important noting there are more than these 3 checkpoints that are present in the cell cycle, but these are the 3 major ones that it's important to be aware of. So in fact, this picture here is showing you the slightly more complicated view of this system. So you can see that at each of these 3 major checkpoints, there are different cyclins that are produced that bind to different kinase molecules that activate different cellular responses. So you can see the situation is much more complex than we have talked about thus far. And you can see here is an even more complicated view of the same example. Again, so now we can see kind of how the cell signaling pathway works. We can see certain molecules inhibit different parts of the cell cycle from going forward. Certain molecules promote growth. So you can see this is actually pretty tightly regulated by cell signaling pathways like we saw in the previous chapter. Now, as you guys may be aware, if the control of the cell cycle is not functioning properly, that is often how cancer results, because cancer results when you have cells that are growing out of control and whose growth is not regulated. So a new class of cancer drugs basically work to inhibit these CDK molecules. They inhibit the kinases that allow the cell cycle to go forward. So this new class of drugs were extracted from some plants found in India, and this drug will basically prevent the transition past these checkpoints because the CDKs cannot become active and cannot phosphorylate whatever their target molecule is. So this is currently being used in melanoma, where a particular cyclin called D2 is overactive due to a high layer of this growth molecule. And these drugs basically inhibit things that allow cancer to grow. So they inhibit things like angiogenesis, which is when cancer cells kind of attract blood vessels towards themselves. It induces programmed cell death, apoptosis, so the cancer cells are more likely to die. It promotes differentiation. One of the qualities of a cancer cell is that they won't be differentiated. They'll be basically like cells that don't have a particular job and therefore are kind of useless to the body. And it will also modify, the transcription and translation of genes that are active in the cancer cells as well. So there are many external and internal factors in cells that will promote the production of cyclin. So in other words, there are factors, external and internal, that also indirectly regulate the cell cycle. So one of these factors is the presence of growth factors. Basically, there has to be an external molecule, usually a hormone, called the growth factor. It's a type of ligand that will stimulate, basically, the division of a cell. So an experiment was done where we had human connective tissue that was cut up and put into a flask, basically meaning that all the cells were separate, and these cells were put into two different containers, one that had a growth factor and one that did not. And in this experiment, we saw that the cells divided only when this growth factor was present, this growth factor called PDGF, which basically showing that if these external signaling molecules aren't present, the cell won't divide. So there is an external force, or sometimes an internal force, that's causing cells to go through the cell cycle. So if we look at the cell type that we just talked about in this experiment in particular, these fibroblast cells, so on their surface they have little receptors. In fact, these receptors are receptor tyrosine kinases, like we talked about in the last unit, that are specific towards this PDGF molecule. And only when this growth factor is present will we get signal transduction that will ultimately result in the formation of these cyclins that will allow the cell to divide. So you can see the cyclin production is dictated by the presence of this external growth factor. So this picture is kind of demonstrating what I just talked about. So basically, when the growth factor is present, these cyclin molecules will be produced, and the cyclin molecules will be able to bind to the kinase, allowing for phosphorylation of some protein complex and allowing the result of passing the checkpoint to occur. So the presence of growth factors is not the only external signal that can control cell growth. So cells also respond to this idea called density-dependent inhibition. So in other words, once cells reach a certain density, they tend to stop dividing, which makes sense, right? Because otherwise you'd end up with cells on top of other cells, and the cells wouldn't be nearly as organized. So basically, when cells become crowded, they stop dividing. And the way this works is cells have certain proteins on their surface that when they get close to a adjoining cell, there will be a receptor on that adjoining cell that will recognize that a cell is right next to them, right? So we have the ligand is present on the membrane of one cell, the receptor is present on the membrane of the other cells, and that will act as a signal to inhibit division. Now, cancer cells do not follow density-dependent inhibition, hence the formation of tumors. So a tumor forms where basically cancer cells have stopped recognizing density-dependent inhibition, divide anyways, and therefore create a lump or a mass of cells, which is what a tumor is. So finally, the last external signal that can stop or promote cell division is what's called Anchorage dependence, and this is the idea that cells generally will only divide when they're attached to something. So they might be attached to a flat external surface, they might be attached to a neighboring cell. If we're growing cells in vitro, they might be attached to a culture dish, but again, without this contact, cells tend not to divide. Again, cancer cells don't necessarily follow this, which means you can often get cancer cells dividing in the bloodstream or when they're not actually anchored to anything. So like I've kind of alluded to several points in this video before this point, is cancer now is the uncontrolled cell division. So at this point, you have a transformed cell that's dividing indefinitely. Now, cells usually only divide 20 to 50 times before that cell undergoes programmed cell death or apoptosis. Cancer cells don't do this. They basically divide indefinitely. They have mutations where they're basically ignoring checkpoints. They don't really follow density-dependent inhibition. They don't follow anchorage dependence. They don't need growth factors to grow, and they do not undergo apoptosis. So basically, they have these mutations that where the checkpoints either not working properly or they're not able to recognize these external factors that usually regulate division, and we get uncontrolled cell growth. Now, as cancer is a really complex disease, there are many, many, many different types of mutations that can cause cancer. However, mutations in two main types of genes are the main types of mutations we see when we're dealing with cancer. So the first type of gene I want to talk about is a proto-oncogene, and a proto-oncogene is basically a gene that stimulates cell division. It promotes cell division. So this might be the gene that codes for a cyclin, right? So that gene is going to code for a cyclin, and the cyclin is going to promote the cell moving through the cell cycle, and thus it's going to promote cell growth. So the normal version of this cyclin gene, or this proto-oncogene, is called the proto-oncogene. However, if that gene was mutated in a way that the cyclin was always produced, that would always be promoting growth, and thus that would be cancer, right? So that mutated version of the proto-oncogene is called an oncogene. So this is the mutated version. Usually we see an increase in activity in the gene where we're getting too much of the protein produced, and thus too much cell growth. Now, some examples we're going to look at are kinases and growth factors. So if you had kinases that were always on and always phosphorylating the next step, we would always get cell division if that was in a growth pathway. And we're also going to look at two proteins called RAS and EGFR. Now, there are many ways a proto-oncogene can be mutated in order to become an oncogene and thus promote too much growth. So I want you to imagine one way would be maybe if the gene was accidentally moved to a new location in the chromosome. Maybe this is due to a mistake in DNA replication, where we get what's called a translocation mutation. And let's say that this proto-oncogene was put next to a promoter, which remember is a DNA sequence that controls when a gene is turned on and off. If it's put next to a really active promoter, this gene is going to be transcribed and translated into a protein too often, producing too much protein. Similarly, you could get this same gene could be copied multiple times with an error in DNA replication, and this would result in too many copies of the gene and thus too much transcription and translation of the protein. Or we could even just change the gene itself and make a version of the protein that's impossible to break down, or a protein that's more active. So these are all different ways that a proto-oncogene could be mutated to become an oncogene. So now let's look specifically at the example RAS that I had mentioned earlier. Now, under normal conditions, RAS is a G protein that when we get this G protein to be activated, it will bind to GDP and thus phosphorylate a protein down the chain in the cell growth pathway and make a protein that promotes the cell cycle. However, if this protein was mutated in a way that it was always active, even if it had not been, even if it was bound to GTP or GDP, that would be an example of a mutation in RAS that would result in cancer, because this pathway would always be turned on and we would always be promoting cell division. So the other type of gene type that you have to be familiar with that's really common in cancer cells is what's called tumor suppressor genes. So these are genes that normally stop the cell cycle. So it might be a gene that inhibits the production of a cyclin, or it might be a gene that breaks down a cyclin, or it might be a gene that prevents a cyclin from binding to its kinase molecule. So if these genes are mutated and they no longer are functional, you're basically not going to have the protein inhibiting the cell cycle anymore, and thus you're going to get uncontrolled growth. So in the case of these mutations, so mutated tumor suppressor genes basically either aren't functioning at all anymore, or they have a much lower functionality, and they're not able to suppress cell growth, which is going to allow for overproduction of cell growth, right? Cells are going to divide too often. So some examples you might have heard of, BRCA1 and BRCA2 are very famous genes that are associated with breast cancer. And P53, I believe, if my facts are correct, this gene is the gene that has mutations, and something like 50% of cancers are due to a mutated P53 gene. This is a gene that produces a protein that usually halts cell growth, and if this gene is mutated, it can't do that job, and cell growth is basically allowed to occur. So let's look at P53 as an example. So you can see in this case, pretty often, well, not super often, but often, we'll get DNA damage. And if you have damaged DNA, obviously you do not want to replicate that cell. So DNA damage will basically kick off a protein kinase phosphorylation chain that will basically activate this protein called P53. And if P53 is active, it will basically create a protein, right? It will act as a transcription factor and will create a protein that stops the cell cycle from happening. However, if there is a mutation and this protein is not made correctly, we're not going to make this protein that inhibits the cell cycle, and therefore cells are going to grow and divide even though they have damaged DNA. So to summarize what we've talked about so far, there are two main gene types that can result in cancer if they're mutated. Proto-oncogenes usually promote growth. So an example of that might be the gene that makes this HER2 receptor on breast cells. So this receptor basically binds to growth factor, which allows the cell to divide. Now, if this proto-oncogene was an oncogene and thus it was overactive, too much of this receptor would be made and the cell would divide too often. The other type of gene that can be mutated in cancer are these tumor suppressor genes. So an example of that might be this RB protein that inhibits the initiation of the cell cycle. But if this protein were mutated, as it might be in cancer, this RB is not going to be active, and therefore the cell cycle is not going to be inhibited, and we're going to get too much division, too much growth. Now, it is important to note that very rarely will one mutation cause a malignant form of cancer. So usually what happens is you get a mutation in one gene, so you lose a tumor suppressor gene function, so you have a mutated tumor suppressor gene, and thus you get overgrowth of cells. So as these cells divide faster and faster, because division is not regulated, cells that accumulate more mutations are also allowed to divide. So this might allow for the formation of another mutation. So maybe you have a proto-oncogene becoming an oncogene. And then the division gets even more rapid and is less controlled. And again, cells acquire more mutations that aren't, these cells with these mutations are allowed to divide. And you get more and more mutations in different genes until eventually you have so many mutations that not only is the cell cycle not regulated, but also the cell can no longer perform its function. So now you have an accumulation of non-functional cells that are eating up nutrients and oxygen from the body instead of those nutrients going towards healthy functional cells. And that's when you get malignant or a tumor that can spread and can ultimately be much more problematic. Finally, just to end this video, I have a couple pictures showing you the stages of mitosis. So this is something we talked about in a previous block, but remember, all of these topics we're talking about, the control of the cell cycle and cancer, all relate back to this basic process of mitosis. So I'm going to leave you with these pictures. You're welcome to look at them if you want a little bit more reminder of how mitosis works, but if not, I'll see you in class.